Hello and welcome to Cellular Respiration, Part 1. Get your popcorn, sit down, and get ready, because this is the good stuff. All right, now with cellular respiration, as we talked about, photosynthesis, we're making the glucose. The plants are, we're not. The plants are making the glucose, and so they make their glucose and we get, we eat. But in the form that the food is in, it's not energy. It has to be converted. So that's what cellular respiration does, is we convert it into usable energy. All right. So with this process here, this is cellular respiration is a process of releasing energy that's contained in organic molecules, which is mainly glucose, in order to do work. Now, this is an example of catabolism. Remember, kata means to break apart. So we are going to break apart this ATP, and when we break it apart, we're going to have energy now available for cellular work. Remember, here it's stored energy, it's potential energy, it's not usable energy. But then once those bonds are broken, then we have energy available. And here's the bond. Here we have 3 phosphates. Here we have ADP is 2 phosphates, and then the phosphate group that we just split off, the inorganic phosphate. Then those are going to be cycled back. Energy is going to come into this. When it comes in, it's going to add that bond back and into ATP, and this just continues to cycle and cycle and cycle. So the process is for making ATP using oxygen if it's available, and this process releases heat. Let's go back here. So this process actually releases heat. Now remember, heat is a low quality energy, but it also releases the heat and free electrons from this. That's what's happening when we're breaking this bond. We're releasing these free electrons and heat. And electrons are a source of kinetic energy. When they're stored within the bonds here, they're potential. Once they're released, they're kinetic energy. If oxygen is present, well, let's look here. First of all, let's look at our structure of ATP. So ATP, adenosine, here's the adenosine right here. It's got a ribose sugar, a 5-carbon sugar, adenine, and triphosphate, 3 phosphate groups. All right, that's ATP. When we go through and break the bond of ATP, so here's ATP, through hydrolysis, we break the bond of ATP, and we're releasing this inorganic phosphate, and now we have ADP. So you can see we went from 3 phosphate groups to 2, and we've released energy. All right, now this word, that's a fancy word, phosphorylation. You should say that, phosphorylation. You should say it around your parents, because they're going to think you're really cool if you say a word like that. But phosphorylation using the free energy, basically that's a fancy way to say we're shoving another phosphate group on using energy. So we're going to make ATP. So when we break it, we have a hydrolysis reaction, but then we go through phosphorylation and we put that back on. So we're going to take this phosphate and we're going to add it back to ADP. Of course, look at here, we have an enzyme that functions to do this, and so we're bound to that active site of the enzyme. We have ADP, that phosphate group, bonds onto here, and we end up with ATP. These PEP and pyruvate, we'll see those coming up. All right, now here's an important question. Is oxygen present? And so here we have glucose coming in, and here's the cytosol of the cell. We have glucose that's coming in, and glucose splits into pyruvate. If it's in the presence of oxygen, then it's going to go into the mitochondria, which we said was the what? Yeah, that's where ATP is made. So it's going to go into the mitochondria, form into acetyl-CoA, and then go into the Krebs cycle. If there's no oxygen, Then it's going to go through the process of making ethanol or lactate, or we call it lactic acid. And uh, so in the presence of oxygen, cellular respiration is going to occur in the mitochondria. Without without oxygen, fermentation, that's what this is, fermentation, that is going to occur in the cytoplasm. That's real important to remember the difference between this. Again, perfect kind of test question is going to make you think about where this is taking place. So if I said something along the lines of that fermentation is taking place, you should be thinking, okay, there's no oxygen and that's happening in the cytoplasm. All right, so here we have photosynthesis and cellular respiration. Like I said, you have to be able to write this balanced equation. So here's photosynthesis, our 6 carbon dioxide plus 6 waters. Now there's no sunlight up here, but there should be. Yields glucose and 6 oxygens, and heat is given off. Now remember this process of conservation of matter, that matter is neither created nor destroyed, it's just transferred. Well, here we go. So now we're taking the glucose and the oxygen, just like I said. Now the products of photosynthesis become the reactants of cellular respiration. Now they intentionally left out sunlight because sunlight's not used in cellular respiration. They didn't want to confuse you. They're just showing you the products and the reactants here. Now you can see the reactants of photosynthesis become the products of cellular respiration. Again, heat is given off and free energy. I'm not sure what that was all about, why it made a strange noise. Okay, now the 3 steps of cellular respiration, and you got to know the steps. If you start very basic, very general, and then we're going to work specific. Step 1 is glycolysis. Step 2 is the Krebs cycle, and step 3 is the electron transport chain. All right, so let's go back here to step 1, glycolysis. This is where we're going to break glucose into 2 molecules of G3P. All organisms can do this process as it occurs in the cytoplasm of the cell. Notice it's out here. It's not in the mitochondria. Very important that you take note of that. So glycolysis, the breakdown of glucose, lysis, we're breaking it apart. We're breaking down the glucose. So glucose is broken down into G3P. Step 2, let me see if I have this. Step 2 is called the Krebs cycle. And this is all about making these electron carriers. See, we're going through this, and electrons are carried via, now notice it's not NADPH, it's NADH. Just remember that the P is in photosynthesis. So NADPH is in photosynthesis, but now we have NADH for the Krebs cycle. All right, so and then we move those electrons into the electron transport chain. We undergo something called oxidative phosphorylation. Okay, this is where those free electrons, these free electrons are going to be used to make more ATP. Now, numbers, notice ATP is formed here, ATP is formed here, and ATP is formed here. That's a lot of ATP. So ATP is formed. This whole process, you got to know the numbers. The whole process yields 38 ATP. 95% of the time, though, there's only 36 produced. So if you remember, anywhere between 36 and 38 ATP are formed as a result of cellular respiration. That's a lot of energy. There's a lot going on in here, but we're making between 36 and 38 ATP. All right, now, so when we're going through, let's go back up here, sorry. All right, so when we're going through the process of glycolysis, this glucose is broken down into 2 molecules of G3P, and each molecule is going to be converted into pyruvate. Okay, so it really should show glucose, G3P, pyruvate. And then the pyruvate is what goes into the mitochondria. If oxygen's present, remember, if it's not present, then you're going to go through fermentation in the cytoplasm. So oxygen has to be present if pyruvate's going to go into the mitochondria, and it has to be a eukaryote. Why? Yep, because prokaryotes don't have organelles, so it's not going to have a mitochondria in order to go through this process. All right, now there's two parts to glycolysis. This first one is called the energy investment phase. So what you can see, we're starting off with glucose, and it's broken down into 2 molecules of G3P. And you can see ATP is used. Now, hexokinase, what do you think that is? That is an enzyme that's going to break glucose down into glucose 6-phosphate. And then you could see phosphoglucoisomerase. You don't have to know those. Just recognize there's enzymes that are catalyzing all of these reactions. Look at this. Phosphofructokinase. Yeah, so fructose. So you can see that we're breaking things down. All right, so in order to break the glucose in half, it's going to take 2 molecules of ATP. Here's one. Here's another one. 1 phosphate's put on each side of the glucose molecule. See, here's a phosphate group that's been added on here, and a phosphate group that's been added on here. This is why where it says 1,6-bisphosphate, the 1,6 is just telling you the number of the carbon that it's attached to. So here's one, here's 2. I'm sorry, 6. You don't have to know that, just kind of a piece of information for you. Now, this makes it, when it's broken in half, it makes it unstable. And glucose breaks in half to make 2 G3P. So glucose is unstable. It breaks in half to make 2, let's go back here, G3P. So ultimately, it's going to split apart, and here's our glyceraldehyde 3-phosphate, G3P, thus the name G3P. All right. Now, so we've used 2 ATPs in this one. Let's look at this. Here's the energy payoff phase. So now we're going to convert those G3Ps that we just made into 2 pyruvates. So this phase is going to end up yielding, so see, you can see here, we're making 2 ATPs here, and we're making 2 ATPs here. And we're making 2 NADHs. NADHs serve the same purpose as the NADPHs in photosynthesis. It's another energy molecule. It's just different. It's like the AA battery versus the 9-volt. And so it's just a different form of energy storage. So this is going to yield 4 ATP, 2 NADPH for all of this per molecule of G3P. The cell pays back the 2 it used for the first part. Remember, we had 2 used right here. And the cell's going to use those, pay back those 2 from the first part. It leaves the cell with a total payoff of 2 ATP because we made 2, but we used 2 before. So we get 2 ATP and then 1 NADH. All right, that's what we call the net gain. Now, remember, this whole process occurs with or without oxygen present. If oxygen is present, it's going to happen in the mitochondria. If oxygen is not present, it's going to happen in the cytoplasm. And all organisms do it as it occurs in the cytoplasm of the cell. That's glycolysis. All organisms do glycolysis. Okay? They all do it. It occurs in the cytoplasm. Whew, that was a tough one. Occurs in the cytoplasm. Now, again, whether or not we're going to have oxygen or not determines what we do with these pyruvates. Do those pyruvates go into the mitochondria? With oxygen, they go into mitochondria. If they're not, no oxygen, then it's going to ferment. It's going to go through fermentation in the cytoplasm. All right, that's all we have for today. Goodbye, folks.